Hello friends, I am Dr. Amit Gangwal, Associate Professor at SVKM Institute of Pharmacy, Dhule. In our ongoing video tutorial series as part of online certificate-based course on AI in pharmaceutical sciences, now we will talk about steps involved in AI-driven drug discovery from similarity calculation point of view or from repurposing point of view. You might be noticing that most of the video are centered around AI application in drug discovery only. Basically, entire operations of pharmaceutical industries are automatic and assembly line driven. So the area which was lagging behind in pharma industry was drug discovery, because still there are lot many diseases for which there is no sure shot treatment. For example, diabetes and obesity, we have to take medicines throughout our life period. If we don't take medicament, we will again become victim of diabetes, obesity, and other type of metabolic disorder. In case of AD, that is Alzheimer disease, or PD, that is Parkinson disease, even there is no sure shot formula to manage these two neurodegenerative disorders, for which patient's life is not as comfortable or as easy as compared to the life of diabetic patient or obesity patient. In case of, I mean, talking about in average terms, after taking medicament, diabetes people can at least have some comfort, but this comfort is not in case of AD and PD patient. If we talk about the disorders of different type, for example, progeria, or for that matter, systemic lupus erythematosus, or for that matter, arthritis, or other type of autoimmune disease. If we talk about the disease that is muscular dystrophy or other rare disorder, if you talk about malaria, AIDS, still there is no sure shot treatment. So this is the area which is lagging behind. So in this particular area, there was lack of vision, lack of automation, lack of collaboration from pharma industry type, from the side of pharma industry. So this is the area where AI has huge potential, and it is creating headlines. For example, Exscientia and in silico medicine is continuously giving drug-like molecule through AI technology to clinical trial. So this is the area where there is need of AI most. So in other area, for example, marketing, manufacturing, QA, QC, there is no huge potential or promising promising leads in terms of AI, because already those areas in pharma industries are far ahead. So more and more videos will be focusing on application of AI in drug discovery. So in this video, we will talk about drug discovery point of view from repurposing in similarity point of view. So repurposing, we all know, to find the off-label use of existing commercially hit blockbuster drug molecule. So you may see in the diagram, there are three circles, and in between them, there are common areas. So in common areas indicate the same drug molecule can be used for treatment of more than one disorder, which may have different or correlated mechanism of action. So what are the initial steps? A compound's drug-like features are related to its chemical feature, such as molecular weight, lipophilicity, and the number of hydrogen bond donor and acceptor. So these are the basic features or the parameter of Lipinski's rule of 5. So if two drug molecules have identical features, so one common notion is that they can have similar pharmacological activity, they can drug the same protein for treatment of disorder. Lipinski's rule of 5 excellently states that a drug-like compound should have not more than 5 hydrogen bond donor, 10 hydrogen bond acceptor, a log P greater than 5, that is a parameter feature related to hydrophobicity, and molecular weight not more than 500. Means all the things are in the multiple of 5. These properties are directly or indirectly encoded in the image and fingerprint-based representations of the chemical structure while training the machine learning model. Here, image means graph neural network or molecular graph. So the first step is to acquire data from Campbell or other type of databases. Compound information on structure, bioactivity, and associated targets is very well organized in databases such as Campbell, PubChem, DrugBank, or Zinc15 for that matter. There are huge type of data sets having information on chemical compounds or biologically active molecule or drug-like molecule or drugs. For the query target, for example, EGFR, whose UniProt ID is P00533, compound data including molecular structure in form of SMILES and bioactivity data are automatically fetched from the Campbell database using Campbell Web Resource Client. Above information is screened for binding assay and IC500 measurements. The dataset is to be formatted and again filtered to remove duplicates and entries with missing value. Only bioactivity value in molar units are kept and converted into PIC50 value. IC50 means half maximal inhibitory concentration. The concentration of the particular chemical which is sufficient to minimize the biological activity by 50%. It shows how much of a particular drug or other material is needed or required to inhibit a given biological activity or process by half. Molecular filtering. ADME criteria and toxicology criteria is one of the most important criteria to remove or to accept or discard the chemical compound in drug discovery marathon. Not all the compounds are perfect starting points for drug development owing to undesirable pharmacokinetic features. It means such compounds are to be discarded from above set for virtual screening. These data sets are visualized using radar plots indicating their ADME properties. So after removing lot many unwanted, problem creating, me too, duplicate and outlier type of data, now it's time to remove unwanted substructures. They may cause mutagenic, reactive or other unwanted pharmacokinetic effects, or that may lead to non-specific interactions with assay. After filtration, for example, you will get only these many compounds for going to next step. So PAINS means pain assay interference compounds. They are a prominent source of false positive in the drug discovery process. So we have to identify them, we have to remove them. In the image which I have taken from the paper of Teach OCDD, it indicates that specific interaction A, B and C. So drug-like molecule is given in the fashion of green circle, which particularly fits the protein binding site on the receptor or jumbo protein pocket. On the other hand, non-specific interaction indicates irregular shape, that is key, which is not perfectly fitting on the log, that is receptor or protein. So this is called pain assay interference compound. We should identify them, we should remove them in early stages of drug discovery. So these are biologically active compounds that pretend as possible drug candidates during the early high volume screening used to search for possible new drugs. Another point is compound similarity can be assessed by comparison measure. So if two compounds are similar, how much similar they are, for that we have one famous formula which is called Tanimoto index. Similarity search can be done in various ways. An RD kit, which is a Python library, a cheminformatic library to implement various steps on the drug-like molecule drugs. An RD kit is one of the popular toolkit that help in calculating similarities of molecules directly. RD kit provides tools for different kinds of similarity search, including Tanimoto, DICE and others. Tanimoto is a very widely used similarity search metric because it incorporates substructure matching. In particular, drug structure or two drug structures are there. So how identical they are, I mean bond by bond or substructure or functional group point of view. So the Tanimoto coefficient is the most popular way to measure the similarity of molecular fingerprinting is by computing the Tanimoto coefficient. As seen in the figure below, the Tanimoto coefficient is the ratio of number of fragment positions shared by the molecules divided by the fragment position shared by either molecule. The Tanimoto score ranges from 0 to 1. 1 means both the molecules are almost identical. 0 means both the molecules are matchless. There is no matching features, or they are not comparable. They are not having something in common. So we have two molecules. You are seeing two molecules, and you see here. So molecule 1, molecule 2. So these particular features, these are the substructures, benzene ring, then this structure. So they are the substructures of each molecule. So this structure is common in both. So we are giving 1. So it is actually, this is called one hot encoding, which indicates one particular feature. If you indicate by 1, 0 means that particular feature is missing. So for example, this feature, so it is present in one molecule number 1, so it is having 1. It is missing in molecule number 2, so it is 0. So this particular feature is not present in molecule 2, so we are writing 0. If see, this portion is available in 1 in 2 also, so 1 1 for each molecule. So this is the formula. A equal to number of bits set in both equal to 3. So this, I mean this feature, okay, 1, then this feature and this feature. So this feature, this feature and this feature, these are common in both the structure. So we are writing number of bits in both common 3. Number of bits set in 1 but not in 2. The feature which are present in 1 but not in 2, they are 2. Number of bits set in 2 but not in 1 equal to 0. This is the formula. So both the molecules are identical only 60%, not 100% or so. Now, what is the meaning of similarity? It is subjective. For me, it can be different. For you, it can be different. So if you talk about only shape, so you see all are square, so they are same. All are circular, I mean circle. Shape is same, but size is different, so they are same. Triangular, the same. For other person, based on color, it can be similar. All are yellow, but shape is different. For other person, pattern, on the basis of pattern, it can be same. It is same, it is same, likewise. So it is subjective. This particular beautiful image I have taken from a video presentation of Dr. Priyanka Banerjee as part of series of lectures on Drug Discovery Marathon 2020 organized by Government of India. So these are the Python codes written in software to identify the similarity between two molecules by fetching those molecules from Campbell database. For example, these are various structures. So for this, we need to calculate similarity using Tanimoto index. So these are the codes written in Python, and they are executed in Jupyter notebook to search molecular similarity. So what is the command, what is the Python code for finding similar molecule in Campbell database? For this, you have to go for this type of coding. So this from rdkit.chem import all chem. rdkit is a cheminformatic library. Compound clustering. So you have lot many compounds, so you can cluster them in different clusters based on some similarity. For example, you have two clusters A and B. So all the molecule in cluster A should have some most striking common similarity. So they are in A. On the other hand, all the member of B cluster should be similar, but member of A should not be similar to member B. Otherwise, it will be difficult to manage the cluster. So there should be more and more similarity in intra cluster member, but there should be more and more differences between the inter cluster members. This is the Excel sheet indicating various chemical compounds in the fashion of Ismail string in terms of clusters. Another example, so maximum common substructures algorithms rank among the most sensitive and correct method to measure structural similarity. Here are two molecules, one is caffeine, one is Viagra. So you can see only the red portion is somewhat similar of the red portion of caffeine is somewhat similar to Viagra. So for this type of hands-on training on finding and judging chemical similarity, you can go to this particular website, that is www.bioconductor.org. Various hands-on training tools they have given to see and learn how to measure the similarity. So there are lot many chemicals. Humans cannot identify what part is similar. Here you have to go for machine learning model to give you ready to use actionable insight for drug discovery. Another example is substructures are very significant in cheminformatics. A substructure is just a part of a molecule, not the entire molecule. For example, the left molecule, that is this one, this portion highlight substructure of a molecule in yellow. This is in yellow. This particular part has been separated and shown to the right. This one. So certain substructures determine how a molecule behave as a drug. Might be possible entire drug is not for active, only this is the pharmacophore. So you will need to find this type of structure in the databases for going for similarity search. It means all the molecules that share the same K substructure are likely to, they are not likely, no guarantee, but yes, they are likely to behave in a similar way. RDKit, which is the cheminformatic library, help in matching substructure between two molecules. With this, we finish about substructure similarities and other related concept.